Thank you, guys. As Francis said, I'm the Research Assistant Professor of Restoration Ecology at UVI. And today I want to talk about harnessing thermal resilience in corals, some of the complexity that lies behind that, and why I think it's research that's critical to any advancements in restoration. I chose the word race because I think we really are trying to race extinction with what's going on with all these new diseases, mass bleaching events, etc. And I think we really need to understand how we increase the thermal resilience of corals that we're utilizing in restoration if we're gonna make a difference and try and protect some of these very rapidly degrading ecosystems. So this graphic behind the slide has some pretty interesting significance. This is the annual global temperatures between 1850 and 2018. And the baseline was the average global temperatures between 1970 and 2000. So the blue represents temperatures that were below that mean, and the red represents temperatures that were above. And the deeper the color, the higher above or below that mean it was. So what's interesting with this graphic is right here, that's about the 1980 mark. And you start to see basically it's all red. So all the global temperatures are succeedingly exceeding the mean that we had from 1970 to 2000. And interestingly enough, in the 80s is when some of the first, we started to observe global mass bleaching events worldwide. Bleaching events is broadly defined when the association between the coral and its algal symbiont breaks down and the symbiont leaves the coral. And this is very important because the algal symbionts provide up to 100% of the nutrition for the coral animal. So once that association is compromised, and the algae leave, you can see the white skeleton underneath, which is why it's called bleaching. And then if things don't revert back to normal and the coral doesn't either repopulate or reacquire symbionts, most of the time they starve to death. So normally a mass bleaching event is associated a couple months later with mass mortality. So this is a similar graphic. That's kind of the same image that was before, but now plotted on a graph from about 1880 to present. And what we see, again, this is the decline of coral cover in the Caribbean since about 1980. Over the last 30, 40 years, the average coral on tropical reefs worldwide has declined about 50 to 75%, and it's been very drastic in the Caribbean. This is a closed basin. We experience higher thermal anomalies, so what the decline in coral cover in the Caribbean has been drastic when you compare it to the rest of the world. This is an interesting review that was recently published. I think it was 2019. And Bruno and colleagues did a meta-analysis where they looked at, I think it was 18 different research studies that looked at 66 marine protected areas, and then they compared them to about 80 non-marine protected areas. And what they were looking at is local stressors and regional stressors as compared to the decline of coral reefs compared to the temperature signal. And what they found is in none of those cases did marine protected areas provide or increase the metrics that they were looking at that are basically what we measure the goods and services of the reef. Increased coral cover, increased rugosity, better fish populations. So the conclusion they drew was that there's been a failure of attempts to manage reefs at the local and regional level. And it's simply because these factors are just swamped by the temperature signal. When we get a mass bleaching event, the significant reductions in coral cover basically wipes out any signal that we have from protection from local and regional. So while those things are important and definitely need to be addressed, it's only buying us time if we don't address the global temperature issue. So if we operate under the optimism that we're going to achieve a reduction in CO2 emissions and actually keep the global temperatures down, there's still things that we need to do with respect to coral restoration efforts. And I choose the term intervention ecology because that refers to when achieving historical states are not possible. And I think most of us agree that we're not going to get back to reefs of the 40s or 50s in the Caribbean. So intervention ecology strives to intervene and help the ecosystem at a different steady state, but where it provides as many of the goods and services that the original pristine ecosystems did. So given the context of global climate change, if you ask me what successful looks like in restoration, I would say that it's not how many corals that we can outplant, but it's about trying to outplant the first coral that's going to survive the next mass bleaching event. And I think, as the title of my talk suggests, I think this is achieved by pursuing research that's aimed at increasing the thermal resilience of the corals that we select and then outplant. So before I talk about some of my research, I want to give you a little bit of background on myself. I was born and raised in Florida. I grew up sailing around the Florida Keys in the Bahamas when I was a kid. Each summer, my grandfather and I would take his little 24-foot sailboat and we'd go down to the Keys, and he always joked that I spent more time in the water than I did on the boat. So some of the reefs in the Keys were like my playground since I was 6 years old. And Carey's Fort was one of my favorites. Back in the 70s, early 80s, it used to be what I think was considered one of the largest stands of Acropora palmata in the Caribbean. I mean, it was just healthy. I guess from my eyes then, it was 100% coral cover. It was like you didn't see anything but coral and fish swimming through it. Well, then in the late 80s, white band disease came and wiped that reef out, and pretty significant restoration efforts in Florida the past decade or more, and we've not been successful bringing this reef or any reefs in Florida back from the state that they're in right now. During my childhood, I also started keeping saltwater aquariums from about 9 years old. It started out just collecting stuff from the seashore, keeping little crabs and fish alive, and then I advanced to corals and then stony corals, so much by the time I was in undergrad, I had my entire apartment filled with aquariums, and I was propagating coral fragments and selling them on the internet. So I paid my entire way through undergrad selling coral fragments. It was pretty profitable. So those are some of the tanks in my apartment during undergrad. And some of the things I observed, I mean, they say the first part of the scientific method is observation. So there's some advantages of being able to live night and day with your organisms and look at them all the time. And this was a pretty unique thing that happened to me September 2001. I was watching TV like I always do. And these aquariums that I had were really dialed in. I was experimenting with microprocessors. I was controlling solar periodicity, lunar periodicity, temperature, all kinds of things that are what we think are the natural cues for coral spawning in the wild. And what happened here was I looked at my tank and a bunch of corals were spawning. But interestingly enough, most of the corals that were spawning were Indo-Pacific in origin. And they were actually spawning the time that Florida corals spawn. So I had my aquarium all dialed in to the solar and temperature cycles of Florida, but yet I had Indo-Pacific corals that were spawning almost exactly out of season of what you would expect. So at this time, it was like, wow, you can actually reset whatever the rhythms are in the corals and kind of manipulate spawning to a different time. And I'm going to circle back around to this at the end of my talk because I think this has some pretty significant implications for how we can manipulate and scale up some of these restoration efforts. It was also during undergrad, I kind of significantly up-leveled my building aquariums. I work for a company that's now called Reef Aquarium Designs, and we built pretty high-end aquariums for hotels, some of the wealthier people on South Beach and Coconut Grove. So it was here I really learned about filtration, pumps. Even though these were display tanks, they were all heavily dominated with corals. So this kind of advanced my expertise in building aquariums and aquaculture systems more. And then I started my master's degree at Mo Marine Laboratory. The year I started, their facility was on Pigeon Key in the Keys, which is that little island under the 7-mile bridge. And this was the year that Mo acquired the property down in Summerlin Key. So my advisor and I, this original culture room was a monkey storage freezer. They bought the lab from Charles River Laboratory, and they used to raise free-range rhesus monkeys out on the mangrove islands for biomedical research. And apparently they were some of the best monkeys in the world because they were free-range, but they started destroying the islands out there, and Marine Patrol and all those said, you got to get out of here. So Mo bought the facility. So that room right there, their first coral culture facility down in Summerlin, we put that together, and then I built a lot of the outside systems. So this was my kind of foray into starting to do more aquaculture-based systems for coral propagation. And then for the next 3 years during my master's, I played a pretty integral role in what was really Mo's pioneering of restoration work. At this time, it was primarily focused on vessel groundings, but it was some of the first work looking at size classes and outplanning and patterns and all that on these areas that were damaged by ships and small vessel groundings in the Keys. So I think this kind of laid the groundwork of, what's been kind of increasing in popularity throughout the Caribbean, throughout the world with respect to restoration. I started my PhD at FIU. I built out their coral culture facility and their marine science building in the North Campus. Started to do a lot of work in captivity with the corals there. And it was during my first year, I got the EPA Star Fellowship to look at the different clades of symbionts that were in Orbicella faviolata and try and tease apart the bleaching mechanisms of those symbionts in association with that host. My supervisor was Dr. Todd Lajeunesse, and we started, this was at the time when the whole systematics of Symbiodiniaceae, which are zooxanthellae, started to get hammered out. And we started to apply high-resolution markers to the group and really tease apart what we now know is a vast array of diversity within the family Symbiodiniaceae. So a little background on how this started. It was since the 60s, it was Bob Trench from Belize and Schoenberg and a couple others that started looking at the symbionts in corals. And up until then, it was thought that it was one pandemic species, Symbiodinium microadriaticum, also the algal symbiont or zooxanthellae, if you will, that's found in Cassiopeia. And Bob started culturing these things and looking at them, and they're not the same. So he, by the early 90s, I think he had described 4 or 5 different species of zooxanthellae in corals. And about that time, Rowan and Knowlton came along and they started applying these really crude molecular genetic techniques. So in the 90s, they were looking at corals, Orbicella in Panama, and they applied a restriction digest enzyme to the large subunit gene. And that basically chops up the DNA and makes a fingerprint. Well, it turns out the large subunit is not a very high resolution marker. It's above the species level, and it's very crude with respect to detections. But interestingly, what they found in their colonies, the Faviolata and it was Montastri at the time, but the Orbicella species complex, is 4 distinct symbiont types. And they term them A, B, C, and D. These things were highly divergent and certainly not the same species. But what was interesting Had they picked any other coral in the Caribbean, they would not have gotten this result. So the choice of hosts that they chose to apply these techniques with kind of led the field of symbiont research down like a dead end, and I'll kind of get into that story. So what the common thinking was that, oh, well, we're finding all this new diversity of symbionts, and there's all this diversity of hosts. And so it looked in the beginning, in the early 90s, like these were open systems. Like, wow, there's hundreds, if not thousands of species of different symbionts with different physiologies. You know, there's a whole bunch of host diversity. So these are open systems. This is great news. But as we did more and more molecular genetic work with higher resolution markers, we started using the ITS, which turns out is a very good species level marker in corals. And we started generating these fingerprints. And really the taxonomy of the group just became an alphabet soup for a while. It was clades and then their fingerprint ID. So things started being named like A1, C1, C3, and it was turning into this nightmare of taxonomy. But what we were finding is that there were all these patterns we were seeing, biogeographic patterns, specific host taxon that you would always find a specific symbiont fingerprint in. And what we were finding as we looked deeper with a higher resolution marker and a higher resolution technique is that there was a lot of host specificity. It didn't look like it was an open system, that these symbiont species were co-evolving with the host phylogenies, and the distributions were not random. So there's definitely biogeographic patterns of the symbiont distributions, also biogeographic patterns. And this was when the picture started to emerge, and it kind of created two different camps. There were the camp that was like, well, you know, it's an open system. This is great. The corals can associate with any zooxanthellae they want. You know, that opens up a whole spectrum of considerations as far as, well, how are they going to adapt to climate change? So this is the pattern that was emerging within both oceans, within all the ocean regions, Indo-Pacific, Indian Ocean, Atlantic. You find a few host generalists. And these things can associate with a bunch of different host taxa. But what's more common is that within each region, you find these host specialists that frequently and only occur with a single host species. And then as we did more and more of this work, we started pulling out certain symbiont species that seem thermally tolerant. So C15 is one, and then D1A, which is going to be the rest of the work that I did in grad school and postdoc, that we finally identified and published that this thing's an invasive species in the Caribbean. So within the clade D, let's see. So one of my first papers was actually testing this in aquariums. So I collected samples from all over the world from public aquariums, SeaWorld, Waikiki, Omaha. There was like 15 different aquariums. So it was to test this whole specificity flexibility concept. Are these open systems? Some of the corals in the aquariums that I had collected samples from, they had been in these public aquariums from up to 15 years, and they were with host taxon that they normally wouldn't come in contact with in the wild. So my thought was, well, if there's going to be switching and shuffling and these are open systems, then when I sample these and do the molecular genetics, it's just going to be a mishmash of symbionts. Turns out it wasn't. It turns out every sample that we processed from an aquarium had the symbiont type that we expected from our wild collections. So corals that were in SeaWorld in Florida, but originally came from Australia, they had the symbionts that they had 15 years ago in Australia. So that was even painting more of a picture of there's a high degree of specificity in these associations. There was one sample that was interesting from SeaWorld. They had the coral ID'd as Acropora cervicornis, but it had an Indo-Pacific symbiont. Turns out when we sequenced the host tissue, it was an Indo-Pacific, it was Acropora formosa, which looks similar to cervicornis, but So in that instance, it was actually the symbiont that helped us ID the host taxa and not the other way around, which is kind of cool. So as more researchers looked, it seemed like a lot of the different species that they were pulling out of these hosts that were in this clade D were thermally tolerant. Each one of these alphanumeric represents a species. These were the ITS2 designations. Several of them have names now. This D1A, this is what is now named Dursdenium trenchi, and it is also very thermally tolerant. So again, Rowan started looking at some of the photophysiologies of members of clade C and clade D, and as you start to elevate temperature, the clade C symbionts, they display that photoinhibition typical of a low temperature, low light symbiont. But the clade D symbionts, no change whatsoever. So their photosystems were chugging away just perfectly at 2 to 3 degrees higher temperatures. So definitely some stress tolerant symbionts within the clade D. And the interesting one I'm going to get to is trenchi, because it has a unique ecology in the Caribbean. But at the time, like I said, this was leading into two different camps. And one of the camps was like, oh, well, coral's adaptive response to climate change is bleaching. So this spawned the whole adaptive bleaching hypothesis, or the ABH. And this was a hypothesis that corals bleach so they can acquire new symbionts. And this actually supported some of the restoration work that was done in the Caribbean in the early days, some of the stuff that Moat did, some of the stuff that was coming out of University of Miami, because one of the researchers there found that after the 97, 98 bleaching event, there was a much higher prevalence of colonies with D than there were before the bleaching event. So they use this as evidence to say, hey, there's these shifts occurring during and after bleaching events to a different symbiont. This is evidence of the adaptive bleaching hypothesis. Well, the problem came down to the resolution that they were looking at. Because if this is the whole switching versus shuffling, if a coral expels its symbionts because of environmental stress and they acquire a new symbiont, well, that's adaptation. And it's basically unlimited in the new combinations or genotypes that can happen in the holobiont. We know that there's thousands, if not tens of thousands of different species. So if you were to say that every host taxa could associate with any one of those species, well, the implications for adaptation are almost endless. However, if a coral can only associate with a few symbionts or one symbiont, and you see this shift, which is basically just a reshuffling of the genotypes that are able to live with that host, well, that's acclimation, and that's much more restricted in the new combinations of host and symbiont that can occur. So this is an important distinction, and this is what I spent my postdoc getting at. Problem comes is in the ITS2 or the ITS1, it's a gene array. So you get thousands of copies of the ITS gene. Well, it turns out that there's different copy numbers between different species of symbionts. So I did these reverse ratios, and the D1 fingerprint, when it gets down to about 20% of the total, you lose it in the fingerprint. Where the C1b, you can see it all the way across. Well, it turns out C1, we found this out much later, has 4 times the copy number of D1. So the resolution of the technique is skewed to depending on how many copy number the ITS2 has. And that's what was misleading some of these adaptation, switching versus shuffling experiments. So I switched to real-time PCR. I developed a quantitative PCR technique to identify D1A. The resolution is down to 1% of the population. So real-time PCR is very high resolution, and I was able to detect D1A all the way down to when it was even less than 1% of the total. So we went back and looked at a lot of our samples with this resolution, and the picture started to become more clear. I started to look at Symbiodinium D background populations in host taxa throughout the Caribbean. I think I processed 400 or 500 samples from the freezer. And these were samples that my advisor had collected when he would just go around the world and do diversity surveys. He would go to a reef, he would take tissue samples of as many corals as he could find, and then he would come back and type them out using denaturing gradient, which is a fingerprinting technique. Again, that lacks resolution to find background populations of symbionts. So I took all those samples, or a lot of those samples from the Caribbean, different depths, multiple reefs, and I started analyzing with both denaturing gradient and real-time PCR. Now, most of these samples, when he would go to reef, it wasn't in response to a bleaching event. It was just during normal times to see what the natural populations had. I analyzed the samples with DGGE to determine the dominant, and then I looked at them with real-time PCR to see if there was any backgrounds of that thermally tolerant D1A in the Caribbean populations. And lo and behold, I found that D1A is present in most, good portion of the coral genera in the Caribbean. But what was interesting is that in all the samples that I looked at, even though there was a pretty high prevalence, it's able to associate with a good diversity of host taxa, the prevalence was really low. So when you averaged it out, in all these samples I did, I was finding D in like 6.3% of the total population. So not the dominant symbiont. What was interesting is the samples that I pulled out of the freezer that Todd had collected in Barbados, pretty much the same diversity of genera, but a much higher prevalence. And I was picking up a lot of these with denaturing gradient as well. So the fingerprinting technique was pulling them out, and we were like, well, what the hell's going on in Barbados? And this is when the story got clear. In Barbados in 2005, they had a pretty drastic bleaching event. And it turns out that the samples that I had pulled from the freezer were collected in July and August. Now, when Todd was there, the reefs weren't bleached. But when we looked back the next month, they bleached pretty severely. Hazel Oxenford, our colleague there, was like, the reefs are bleaching like crazy. So when we went back and looked at the temperature, what was happening is those reefs had been experiencing almost 2-degree higher temperature for months. So for months before we even sampled. So what was going on was the host was even tracking that stress before it was visible to the naked eye. So the corals hadn't bleached yet, but the symbiont communities within those corals were already starting to change, with D1A increasing in prevalence in those corals. These are my real-time PCR. In almost every species, there was an increase in D1A from pre-bleaching to bleaching and recovery. So on average, it was in 22% of the population, and then when we looked at it after, it was like 62%. So almost a threefold increase in prevalence in these corals that were experiencing bleaching. And it did protect a lot of the corals from bleaching. So we went back and looked, the pre-bleaching from 2 different depths, you don't see hardly any D1A fingerprint. And it was there, but it hadn't exceeded the 20% mark where you could detect it with fingerprinting. Well, after, if we looked at the normally pigmented, so the ones that appeared to survive bleaching, they were chock full of D1A. The corals that were severely bleached, very little D1A. So the D1A symbiont did seem to provide protection to the animal. So one thing about the ecology of Symbiodinium and D in the Caribbean is that it's unique. So we find different D species. I think right now there's 13 putatively described species in the world. There's only one out of all the fingerprints we would pull out, and we did almost 4,000 samples. It was the same denaturing gradient fingerprint. So this turns out to be really interesting. D1A, which is now called Durasdenium trenchi, it's the only species within clade D within the Caribbean that we found so far. And what's interesting is it's not recognized as the dominant symbiont of any coral taxa that's not stressed. In other words, if you go to a reef and the reef's doesn't appear stressed, you will not find D1A as the dominant symbiont unless it's residing there from a previous stress event. Because it turns out it takes almost 2 years for the populations to revert back to normal. And this is what was seen in Florida after the 97-98 El Nino. We saw this rise and fall of trenchi over about 2 years. And we went back 2 years after the Barbados bleaching event, and most of those colonies had reverted back to their normal symbiont populations. But we did find that D1A can become opportunistically dominant during a stress event. So these are some of the data. This is the 2005 Barbados. 2 years later, we went back, and there's very little D1A in those colonies. This was colleagues that did the same thing after the 97-98 El Nino. D1A is the yellow, and you see after 2 years, it starts to revert back to the normal native symbiont populations. So what are these guys doing in the coral? I mean, we were really asking the question, well, what's the ecological relevance of hosting a symbiont in less than 1% of the total? I mean, is it just moonlighting there? What's going on? So all that data led to the publication Outbreak and Persistence of an Opportunistic Symbiont during the 2005 mass bleaching event. But I wanted to go further with that because the picture was becoming clear that D1A in the Caribbean was not like D1A in the rest of the world. So I went to Puerto Morelos, Mexico for my postdoc. You'll see a common theme here. I got there, I built out their seawater systems. It's pretty state-of-the-art ocean acidification setup. 8 different reservoirs, 4 different PCO2s. We can manipulate temperature, light, and ocean acidification in those systems. This is just showing the highly different photophysiologies between different symbionts. This was some of my advisor's work, Bob Trench. And then this one is cool because people were starting to look at, okay, well, what are the potential trade-offs of hosting a thermally tolerant symbiont? Now, this is D from the Pacific versus C. And what Little et al. Found was that it does decrease, having the thermally tolerant symbiont seemed to decrease growth rates, although these growth rates were taken by imagery, so it was just tissue and not actually calcification. So when I got to Mexico, it was 2008, I believe. Bill Fitt had been doing seasonal surveys on the reefs in Puerto Morella, and in 2005, the same year Barbados bleached, the Mexican reefs bleached pretty bad. And what he saw was this rise, the brown being D1A. These are the fingerprints. This is the denaturant gradient. So again, what he detected here meant that D1A was rising to at least above 20% of the population. And then over the 2-year period, it kind of faded out. When I got to Mexico in 2009, we had another pretty serious bleaching event. And I saw this rise of D1A. And it didn't revert back though. By the time I left in 2012, a lot of the colonies there were still dominated by trenchi. And I know Bill's lab went back there in 2018, and there was still a lot of trenchi. So what's going on? Is this thing here to stay? Is it conferring thermal tolerance to the host, and is that a good thing? Well, all these papers were coming out suggesting that it is a good thing. This was support of the adaptive bleaching hypothesis. There was this nugget of hope paper, clade D symbionts, nugget of hope. So there was all these things, and it was also starting to drive restoration. I know Andrew Baker and some of his colleagues got some funding in Miami to culture D1A and then take it out and try and seed the reefs with it. So they were actually trying to promote D1A and host taxon, thinking that was going to increase thermal tolerance. Well, all of these papers said something to the effect of, but we don't know what the physiological trade-offs may be. So this is what I decided to do. I decided to look at calcification rates in corals, specifically Orbicella faviolata, hosting its native symbionts, which on the Mexican reefs, it can host 3 different symbionts. It commonly hosts an A type, a B type, and a C type. But then you can find D1A if you look hard enough. And lo and behold, what I found was that Orbicellas that are hosting D1A calcify about 50% less. So these are the 3 native symbiont types, A3, it's either B1 or B17. There's 2 different B symbionts it can have, or it's C7. So those are the native symbionts in Orbicella on Mexican reefs. And I went out and found colonies with all 3 different symbiont types, and then colonies dominated with Trenchi. And I measured instantaneous rates of calcification over a range of temperatures, because I thought, well, maybe it behaves better at a higher temperature. Turns out it doesn't. Turns out that they calcify on average 50% less if they have that symbiont. So that's a significant trade-off for thermal tolerance, is to reduce your calcification levels by that much. So I decided to look at photophysiologies. When I measured oxygen evolution, and I also did this over a range of temperatures, you look at Pmax between D1A and then 2 of the native symbionts, there's absolutely no different. So the algal symbiont is behaving like an algae. It's photosynthesizing, it's producing oxygen. So what's going on? Why is it not contributing to calcification like the other symbionts are? So this is just a graphic showing that at the same levels of Pmax, you get about a 50% reduction in calcification as compared to the native symbionts. So we came up with a model of photosynthetically driven calcification. So in natural symbioses, the symbionts are providing 2 things to drive mitochondrial output, and this is what drives calcification. They're contributing oxygen, and then they're contributing glycerol. And this is what helps. Calcification rates are typically 4 to 10 times faster in the light than they are in the dark. And that's the result of this. So we hypothesized that the opportunistic symbioses, we know that it was creating the same amount of oxygen, but we hypothesized that it wasn't either making or translocating glycerol. And it turns out one of my colleagues showed this recently. So when I presented this at the Euro ISRS, we termed it the selfish symbiont hypothesis. And we were kind of counteracting the adaptive bleaching hypothesis. So one of my colleagues actually looked with some isotope work, looked at carbon translocation in Orbicella. And again, he found the same thing I did. The PDR ratios were identical between Trenchi and the native B1 symbiont. C13 in the alga and the host tissues was pretty much identical. So these things were creating glycerol, but what was happening is it wasn't making its way to the skeleton. So that kind of supported our photosynthetically driven model, whereby it was the lack of glycerol translocation that was reducing calcification. So with that, that led to the paper. It allowed us to call this thing not only an introduced species, but an invasive species, because it was significantly affecting the physiology of the host. And what was most interesting, we started looking at even higher resolution genetic markers. And we went to Palau, and you could do a 50-meter transect, and we would find 50 to 100 different genotypes of Trenchi on a 50-meter transect. I think I did over 4,000 samples from the greater Caribbean, and we found that there's 2 clones of Trenchi in the Caribbean. So that's what blew it apart for us, was the physiology, the calcification work, and then seeing that, wow, this thing was definitely introduced into the Caribbean, and it was probably pretty recently. Two clones is all we found. And I know Todd has continued this work, and I don't think he's found anything more. So this thing was introduced recently, and it does confer thermal tolerance, but at a pretty high cost to the host. This is just a graphic of how most reefs in the Caribbean and actually the world are right above the stasis level. So what is a reef? It's when net accretion exceeds net erosion, you get the formation of a reef. Well, what's going to happen if trenchai becomes fixed in the Caribbean populations and reduces calcification of the reef building corals by 50%? You're going to see this shift of reefs to net erosion. And that's barring all other factors like the disease reducing coral coverage and all the other stressors that are bringing that down. While I was in Mexico, Anya and I did some side projects. We were looking at the effects of temperature, PCO2, ultraviolet radiation, and symbiont species on larval survival and growth. So this was also Orbicella. Last year I was there, we collected gametes, fertilized them, settled the polyps out, and then inoculated them with different cultures of symbiont. This experiment ultimately got destroyed by a hurricane, so we don't have any publications or results, but what I did find was some cool stuff again with trenchai. During our infections, I was following them using an epifluorescence scope, so I could count how many algal cells had made it in to the aposymbiotic polyp. And it was pretty neat. Again, when I compared the two native symbionts of Orbicella with trenchai, so this is C7, that's one of the native symbionts. Had 1 or 2 algal cells 48 hours after inoculation. 96 hours, 15 to 20, 144 hours, approaching 50 to 100. Saw the same thing with A3. This is Symbiodinium fiti. 48 hours, you get a couple algal cells that made their way in. 96 hours, get a few more. 144 hours, you're still somewhere in the range to 50 to 100. With trenchai, the infection rates were similar, but then the proliferation rates went through the roof. 48 hours, 96 hours, and by 144 hours, I couldn't even count them anymore. So this thing makes its way in to newly settled planula quickly, and if available, it proliferates a lot more quickly than native symbionts. So some of the questions that immediately arise to me are, well, with what I saw it doing to calcification in the adult colonies, is it inhibiting that initial growth in juveniles and newly settled polyps? I don't know. That's one of the things I'm going to continue looking at. So this all led to a bunch more research that we've been doing in Palau for the last 5 years. We think that this is where trenchai originated. It associates with a lot of corals in Palau. There's lots of different genotypes of this species, and it does confer thermal tolerance to those corals. What's different though, is if you measure rates of calcification in those corals, they're normal. So we think that D1A is from the Pacific, probably somewhere around that region, and it's co-involved with those corals for millions of years, which is why you don't see that physiological trade-off. This is just a bunch of the experiments. We did a whole bunch of reciprocal transplants. These rock island corals in these bays, they get up to 34 to 35 degrees Celsius and they're fine. And most of the corals in the rock islands are dominated with trenchai, but their calcification rates are normal. Got some papers coming out recently looking at the different functional traits among the symbionts there, including the thermally resilient symbionts. And this was my last 2 years. I was hired as the coral aquaculture supervisor for the Nature Conservancy, and it was basically to come in and build their coral innovation hub on St. Croix. They have 27 acres right down from Christiansted here, and we built out basically a laboratory and coral aquaculture systems using recycled shipping containers. This is just some of the lab. I built some interim systems when I got there so we could start doing work. And this was the installation of the formal lab. It's 24 raceways. 3 separate systems. Right now, they're completely closed systems. We've been working on getting a seawater intake permit because ideally the way to do it is to have it either an open system or a semi-open system.
Did you start building right after the hurricane or right before?
Right after. I wasn't there for these hurricanes. So this most recently led me to start my old business back up with Matt. Matt's got a significant background in building large desalination plants all over the Caribbean. So our idea is to compartmentalize and make these things modular so we can ship them all over the world. Basically, take an empty shipping container, build out all the filtration inside, ship these to wherever they need to be. The organization or lab that's there does all the site work, concrete pouring, pedestals and all that. Put the filtration container in place, do all the hookups, in a relatively short period of time have the ability to propagate corals. So I just got this position at the University of Virgin Islands. As I said, I'm going to be the new research professor of restoration ecology there. So a lot of my research will be focused on harnessing thermal resilience in corals. And some of the things that I'm working on now and will be working on soon, I'll talk briefly about. The microfragmentation technique. I've been wanting to do these experiments for a while. I have some preliminary data from the Bahamas from wounding experiments I was doing to suggest that microfragmentation is not really increasing growth per se. What I think it's doing is I think the coral's reallocating its energy into healing its tissue, and I think that's at the expense of calcification. We did these wounding experiments years ago in the Bahamas where we would simulate a parrotfish bite and then look at recovery of that wound in marine protected areas and outside to see if there was any difference. But what we found was that the coral basically it goes down to almost 0% calcification until that wound's healed, and it doesn't make its way back up to 100% calcification for almost 6 months. And I don't know if it progresses longer than that because we ended our experiments at 6 months. But if you read the papers on microfragmentation, all of them use imagery to quantify quote unquote growth. So I'm going to take a harder look at this, and I want to see if the trade-off is indeed reduced calcification, and then also if there's implications to do with fecundity, because we know when corals are stressed drastically that they can actually consume their gametes. So there may be some other negative effects of microfragmentation on the parent or the donor colonies. So this is something that I'm going to investigate at UVI. I'm also working on a model of endolithic calcification. I've got some preliminary evidence to suggest that the endoliths in the coral skeleton actually contribute to calcification. I made these upside down chambers where I had access to the skeleton, and then I actually added some, it wasn't chloramphenicol, it was another photosynthetic inhibitor. And I showed with dyes that the inhibitor was not making its way to the outside. So the tissue was isolated in a water bath underneath, and then the column gave me access to the skeleton. Well, when I added the photosynthetic inhibitor to the skeleton with the endoliths in it, it reduced calcification in Orbicella by about 40%. So I need to follow this up. I did some experiments in Palau because what I need to make sure is that the photosynthetic inhibitor is not making its way to the symbionts. So I built some upside down chambers where I could pamphlerometer the tissue of the coral while I was adding the photosynthetic inhibitor to the skeleton, but I have to take those experiments a little bit further. One of the things I'll be doing next month is looking at the gamete quality from Faviolata from parent colonies that host trenchi. So the one trade-off that I found was reduced calcification. We've got some preliminary evidence that the gametes that come from parent colonies that are dominated by trenchi have a less optimal lipid profile. So obviously lipids are the stores for the egg, for the planula. So it's very important. We found, and this is in a limited number of samples, but we found that total lipid concentrations in eggs from corals with trench eye are reduced. And then also the lipid classes change. And this TAG, that's one of the important lipids for energy. And you see a drastic reduction in colonies that the eggs came from parents that had trench eye. So we want to follow up on that. I'm also experimenting. We've got some experiments in process right now with one of my interns at TNC. We've inoculated all these different polyps with different strains of symbiont to see if indeed we can enhance both growth and survivorship in these early stages. And this was spawned by the stuff I did with Anya, seeing the uptake and increased prevalence or proliferation of trench eye in those planula. If that gets in and it's reducing growth rates in larvae, well, then I would argue that facilitated sexual reproduction restoration efforts should take consideration of what symbionts they're inoculating their larvae with. Some organizations just allow them to uptake naturally from whatever the incoming seawater is. But if we find out that you can give them a jump start by either giving them their native symbiont or something that's better than trench eye, then that may become important for any of these restoration efforts that are doing sexual reproduction. We've also just submitted a big NSF proposal, tons of colleagues in this. We're going to try and create a global phenomic network for reef building corals, which is basically building a cost-effective benchtop system that allows non-experts to do thermal challenges with corals in a non-destructive way. So we would use fluorometry, we would use spectroscopy, we would look at reflectance as a metric for bleaching, and then also tease apart what's going on in the photosystems during a 14-day temperature experiment. So it would allow restoration projects, organizations to be able to tease apart, well, do some of my genotypes show thermal resilience? Can I test that easy without doing a whole bunch of, post-processing and, a lot of molecular genetic stuff? Can I do that easy? And then can we share all this information? David Kramer from Michigan, he developed this photosync for farmers, and it's taken off in agriculture. It's basically a handheld fluorometer that farmers can do real quick and dirty analyses on their crops with, but then that information automatically gets uploaded to a cloud-based database, and it shares that information all over the world. So we want to copy something like that with our thermal fragment analyzer. This is some of the research that I'm working on now. Some of the Australians were actually doing these experiments. They call it assisted evolution. You may have heard of it. But they're focusing on the algal symbionts. Generation times of the symbionts are every other day. Generation times of the corals are, pretty widespread. So they've been doing these ratchet experiments, challenging cultures to elevated temperatures and then trying to create and pull out mutants. And some of the work's really cool. They're creating mutant strains that are 1 to 2 degrees more thermally tolerant than the wild type. My colleague at University of Delaware has done this, and we've actually created a breviolum, which is the symbiont of fire coral, I think. Anyway, it's one of the B types. And we've got a 2 degrees more thermally tolerant wild type. So he's going to bring that down hopefully after spawning this year, and we're going to see if we can get it into some of the larvae and then see if it persists, and then see if that thermal tolerance is conveyed to the holobiont, or whether it's just transient and it goes away. So these are the ratchet experiments. We're going to infect them into larvae. Obviously, if we can create a subset of thermally tolerant holobionts, that'll be great, but what will be even cooler is if we can raise those corals to reproductive age, and then they come back and produce gametes that also have some type of thermal tolerance. That would complete the cycle, and that would start to increase thermal resilience in restoration outplanted corals. So now the race. Well, the problem with coral spawning is we only get one shot a year. But I told you what I saw in my aquariums, and there's recently been some publications related to that. What if we could get the corals to spawn every year? And this is possible. We know that we can manipulate their spawning cycles. So just set up staggered systems that are getting gametes every year out of these corals, and that would significantly increase the rate at which we could scale some of these technologies. Jamie Craig's just published, I think it was last year, they did something similar in the Horniman Museum. They brought corals in a couple years, it allowed them to reset whatever their rhythms were, and they got out of season spawning in captivity. So serious implications for restoration, to be able to have the ability to have gametes whenever you want. There's also been some recent advances with cryopreservation. So we're finding ways to cryopreserve both the eggs and the sperm. So if you could have, material on hand and thaw it out when you need it, that would also increase the rate, I think, at which we could scale some of these restoration projects. So this is basically some of the papers that are coming out looking at 10 plus years of restoration in Florida with no significant progress, no significant success stories. So this is why I think we need to step back from the whole scaling and the whole, well, how many corals we can plant, and really start to get down to some of the applied and experimental work to figure out how we increase the resilience of the corals that we're planting. So I'm going to circle all the way back around. To me, what restoration looks like, it's about putting that first coral out there that's going to survive the next bleaching event, and then having that coral reproduce, and then having those larvae settle and grow up into corals that can survive the next bleaching event. I think that's going to be the best way that we can contribute to protecting these ecosystems with our restoration efforts. And that's it. Thank you. Anybody have any questions? It's all over Florida, yeah. And a lot of the corals, yes. We found trenchi in almost everything. It's prevalent in the Orbicella, but you'll find it in the acroporids. There's very few species in the Caribbean that we don't find it in, especially when they're stressed. There's a bunch of corals. A publication came out last month looking at these coral assemblages in Miami Harbor. It's a pretty turbid, heavily impacted site. Everything in there is full of trenchi. So those corals are stressed, chronically stressed. And what's interesting, I went out to the Flower Garden banks in the middle of the Gulf. Those reefs are at like 100, 120 feet, and they're beautiful. There's no signs of disease. And they had a token acropora there. It was on their newsletter for like 2 years until the thing finally died. And when I went out there and did a diversity survey, everything had what it should have, except for that acropora had trenchi. So, I mean, you look at a coral that's living way beyond its range, chronically stressed, and the symbiont it's hosting is the opportunistic one in the Caribbean.
Are there any theories of how trenchi came to the Caribbean?
No. No theories, but based on the lack of diversity here, I mean, we think that it was recent. I mean, even in a few thousand years, this thing should have diversified more than it has. And we've done, I don't think it's a lack of sampling. When I left the lab, we were over 4,000 samples, and we were still only finding 2 clones. So that's pretty impressive when you go to plow and there's 50 clones or 100 clones in a 50-meter transect. And Phuket is the same way. You do a 50-meter transect in there and you find tons of different genotypes of trenchi. So what's going on in the Caribbean with this thing is definitely unique. Yes?
You talk about trade-offs, and then you mentioned, that you're doing work on trying to maybe get corals to spawn more frequently. But
Yes.
So they could make that
No,
coral.
good point. It wouldn't be the same coral spawning multiple times a year. It would be that, so say you took one colony and created 12 fragments, and then each system was staggered. So it wouldn't be the same no, that you couldn't produce more than one gamete cycle a year, but you could set up systems where those time frames were staggered. So that's the way I was envisioning that.
And then my other question is also about trade-offs. You talked about looking at OFAB with the declayed and showing a 50% reduction in calcification, but the same thing happens when a coral is bleached, right? It's not getting the energy that it normally does to.
Right.
So is it really a trade-off? Because if the coral didn't have the declayed, it would be more prone to bleaching, and the bleaching is going to slow down growth and calcification as well, and they can stay in that stress mode for months and months and months.
Yep. So yeah, that's.
Is your study looking at bleached or a coral with the declayed and a healthy colony without it?
Yeah, I have some of that.
Or was it a bleached colony with the declayed?
I did that. I bleached some of these colonies and then watched the return of calcification. And I can't remember, it was in a few months, even though it was visibly bleached, it had repopulated with enough native symbionts that it exceeded the normal calcification rate of fully pigmented trench-eye colonies. So I did look at that, but you're right, yeah. I mean, and that's still the argument. Well, is it better to be alive and not calcifying or bleached and dead? I mean, I just think we need to be cautious because, and I know that a lot of restoration programs are focusing on the thermal tolerance of the symbiont without the understanding that maybe trench eye is not the symbiont to be utilizing. I mean, there's other symbionts in the Caribbean. A3, Fiti is pretty thermally tolerant. So I think there's other choices if we're going to start to do these manipulative kind of experiments. Keep going.
You were also saying that when you looked at Palau, that almost all the corals there had declayed.
In the Rock Islands, yes.
In the Rock Islands. But were most of those corals like aquaculture? Like I'm wondering if the work you've done with the declayed in the Caribbean was with OFAB.
Yeah, in the Rock Islands, there's only one or two acroporids. It's mostly boulder corals and whatnot. Boulder corals, and then there's a lot of the foliose corals. I mean, I don't know, there's the species names there are insane. They look like agaricids almost. But very high nutrients in there, very high temperature. What we found out is that the coral's not maintaining its calcification rates through heterotrophy. It's actually coming from the symbionts. So that was kind of surprising to us. We thought, oh, well, maybe they're better fed and that's how they're keeping up with it, but that's not the case. Yes.
So you said that those temperatures are 35-36 Celsius there. That's constant temperature, or that's?
In the Rock Islands, it's pretty hot all the time. Yeah. There's not a lot of flushing. I mean, you get these closed kind of basins, and it heats up in there.
Did you test those for increased temperature as well?
They are thermally tolerant, yes. They have trench eye. They can withstand higher temperatures. The conspecific host taxa that's out on the outer reefs has a whole different assemblage of symbionts.
You think that the presence of that in the Pacific is more because of prolonged stressing events in the past? Is there record of prolonged stressor events in the past?
No, I think it's more stressful in the Caribbean because we are a closed basin. And I think that's why you see a higher decline and a higher prevalence of disease here. I mean, it's interesting to me that all these coral diseases started being observed in the early 80s. Well, that's when global bleaching events started happening. So I think it's kind of like the model system. I mean, there's disease all around you right now, but if your immune system's in good shape and you're not stressed, well, you don't get sick. And that, you see a lot of disease pop up after bleaching events. So I think the But there are other species of thermally tolerant symbionts. It's not just members of Durasdinium, nor is it just Trenchi. I mean, there's some rugged symbionts out there. Anything else?
You know, we're all here working in restoration, and I have to say some of your, you know, studies are just really like, well, what are we here for anymore? Like, so what would be your take-home message to little islands like us that don't have all the technology to be in the lab looking at, you know, the symbionts and
so I think
like,
that,
are we doing the right thing with, like, I mean, because I feel like it's better to be doing something like, and at least like this little band-aid of trying to preserve some genetic diversity out there.
Absolutely.
We have more information from
absolutely.
more sophisticated studies, but at the same time.
No, what you're doing is not wasting your time. I feel like a lot of the organizations just play a numbers game. And it's like, well, if we can plant 30,000 corals, and you know, I think this was one of my problems with TNC. It was always about more numbers, more numbers, more numbers. But when we would do the monitoring on survivorship, everything we're putting out there is dying. So it's not about, clearly we're not gonna fix this problem by just putting out more numbers. But I think one of the low tech things that can be done is selective breeding. I mean, it's been done in agriculture for thousands of years. You take a plant that has a characteristic and you take another one and you cross them. And that's very easy to do. I mean, you don't need the molecular analyses or anything like that. If you observe a phenotype that seems thermally tolerant and you observe a phenotype over here that seems to have disease resistance, but they're far enough apart that they would never meet, well, bring them together. And, you know, I think facilitated sexual reproduction is one of the big directions that this work needs to go. I don't think that we're ever going to succeed by playing the numbers game.
You said in one of the studies that you were concerned with microfragmenting, it really isn't the like traditional growth, it's more like the healing of tissue and not so much calcification. But I went to a coral restoration workshop at home with Dr. Vaughn, and you know, we saw corals, brain corals that he had put, you know, together that fused, and in 9 months we're actually now producing gametes. So I would say that like it's calcifying enough because the coral isn't coral reproduction.
So
more on size, not so much age. So like if it's reaching these sizes,
right.
You know,
Well, they made the comparison that those corals, I thought it was 2 years, but those corals were reproductively viable when it would take a normal coral, you know, 10 plus years. The problem with that comparison is that the host colony that they got those microfragments from was probably reproductively viable anyway. So the question to me becomes, well, when you took those samples, did you reduce the fecundity of the host colony? Because there's a lot of unanswered questions. I mean, I'm not bashing all of it. I just think they do there's a bunch of arm waving about this exponential growth. Well, maybe on the tissue side of it, but definitely not on the calcification side. So, you know, I think we need to look at it more rigorously and say, well, is that an acceptable trade-off? If we're going to grow tissue, you know, 4 times faster, but we're going to reduce calcification for 6+ months, I mean, is that the way you want to go? Because, you know, at some level, it's not the tissue out there that's protecting from waves and storms, it's the skeleton. A lot of unanswered questions, and I
mean, I know like my Palmadas is much better outplanted than the cervicornis.
Yeah.
But my cervicornis do really well in our nursery, and we've had some studies done where we introduced white band disease to some of the different genotypes, and we found genotypes that with white band directly contact is not getting it.
Yeah.
So, you know, we like to do similar things with, you know, bleaching.
Yeah.
We haven't had any real bleaching observations in our nursery, but you know, some of our corals are getting so big that I do think they're reproductively mature. And for me, it's almost like I'd rather keep them a
Yeah.
just an evolution where we get the genotype that's resistant to disease with one that's thermally tolerant, and
Yeah.
those are going to have better chances of larvae settling in the outplant.
Yeah. Now, I think there needs to be much more focus on sexual reproduction rather than just fragmenting.
I think you're here to consult on what we can do
Yep.
at a smaller scale. I mean, obviously, we're not going to be creating anything like the Virgin Islands.
Yep.
Certainly, country, wasn't it?
Yeah, you can stack them if you need to.
We're starting and then moving forward, hopefully next year. So we have 2 persons going to Dominican Republic this week to learn about sexual reproduction. So that's going to help us maybe in the future to actually.
Have you visited the site where we have been given the space?
I have, yeah. Yep.
That was my house.
That was your house? Oh, that's awesome.
I'm like super excited that this is what it's going to become. Like, if we can go to anything, I'm like, yay.
Oh, that's awesome. Yeah, we checked out the lagoon there for installation of a seawater intake, and then took measurements of the place there. I mean, it's actually perfect. It's a really suitable site. Just got to take some of the trees back. There's no light in there right now.
We'll take an extra the almond tree. Yeah, and the idea is that this is one of our projects from the Swiss Corporation. So we're hoping to have this up and running by the end of the year. And this is part of our effort to fight stony bull tissue loss disease here on the island, which is something that has frustrated many of us. So it's really, so we're looking forward to this project. Maybe hopefully it's not going to be like a 1-2 year project, but it's going to be a long project with other organizations, not just the RFP, but everyone on the island. We hope that this project will replicate in other islands in other parts of the Oregon. So this is just a start.
Awesome.
So thank you so much, Robin.
Thank you.
It's great to have you here.
Thanks.
Doing that.
It's great to be here.